Hi, this is Dr. Prasad Pradil. I'm continuing class series of Biology for Engineers. This is for BBOK407, that is other than computer science stream students, mainly the electronics, mechanical and civil engineering students. For them, the part 2, part 1 almost half module completed. Now this is part 2, another half, almost half I'm covering. So those who are watching my videos, please do subscribe and support. With your subscription, that is the only support that I am expecting from you. As such, the study material, this PPT PDF, I will be posting under the description box, and also within a few days I will be posting once the video is uploaded and started viewing. Okay, so this is my YouTube channel, my intuition 4865. Please watch my videos only in this particular YouTube channel. Okay, so here I am continuing the introduction to biology. This is again I am repeating. This is BBOK407. The syllabus, it is for electronics, mechanical and civil engineering streams for those students who are joined to Vishweshwara Technological University in the academic year 2022. Okay, so continuing. So this is the syllabus of introduction to biology module 1. And the course outcome are mentioned here because once you are to the outcome based syllabus, you should know what is the course outcome. So course outcome is mentioned here. 4 course outcomes are there. You need to go through that. Even teachers also should be aware of that. Based on the course outcome only, the question paper will be prepared. So I have framed one question for this. Provide an overview of biomolecules highlighting the properties and functions of carbohydrates, nucleic acids, proteins and lipids. Explain their significance in cellular process connected to course outcome 1 with the level of question as 3, revised Bloom's taxonomy level question 3, and carries 10 marks. So the answer scheme also given here, that is overview of biomolecules carry 2 marks, highlighting the properties, functions and significance of carbohydrates, nucleic acids, proteins and lipids, 2 marks each comes. It comes 8 marks. So 4 are the biomolecule, carbohydrate, nucleic acid, protein and lipid. So 2 marks each of the 4, it becomes 8 marks. Okay, so we'll start the answer scheme for that, or answer points for that. Okay, so for 2 marks overview of biomolecules. Biomolecules are, they are the organic molecules which are very essential for life. It is found in all living organisms. They are actually primarily composed of carbon, hydrogen, oxygen, nitrogen and phosphorus. So the 4 major categories of biomolecules, they are the carbohydrates, nucleic acid, proteins and lipids are the major things. Then other biomolecules like enzymes, hormones, vitamins are also there. So each of these plays a very crucial role in various cellular processes, contributing to the structure, function and regulations of the body's tissue and organs. Now we move on to the first biomolecule, that is carbohydrates. Properties, function and the significance is given here point wise. Okay, so properties, the first point about the structure only, the composition. It is composed of carbon, hydrogen and oxygen, typically in the ratio of carbon, hydrogen and oxygen in the ratio of 1 to 2 to 1. Okay, so that 1 carbon, 2 hydrogen and 1 oxygen, it gets repeats n times. That is the structure of the general structure of the carbohydrates. So these carbohydrates, they are actually simple sugars. It includes simple sugars like monosaccharides, which are glucose, fructose, disaccharides like sucrose, lactose, polysaccharides like starch, glycogen, cellulose, etc. The primary function of carbohydrate are to become the source of energy for the body. Okay, so the simple sugars provide immediate energy, while the other polysaccharides like the glycogen, they serve as energy storage. Later stage it will release the energy. The structural carbohydrates such as cellulose, it provides support in plant walls. Okay, so significance in cellular process. Carbohydrates are very crucial in cellular respiration, where glucose is broken down to produce ATP, adenosine triphosphate, that is ATP, and the energy that is called as the ATP is called as the energy currency of the cell. So they also play a very role in cell recognition and the signal process. Then comes the nucleic acid. Nucleic acids properties includes that of DNA and RNA. They are composed of nucleotides, monomers, which consist of the phosphate group, the sugar, which is deoxyribose, that is in the case of DNA, and ribose in case of RNA. And then there is a nitrogenous base, consists of adenine, thymine, cytosine, guanine in case of DNA, adenine, uracil, cytosine, and guanine in case of RNA. So the thymine is replaced by uracil in case of DNA to RNA. Now the functions. DNA stores genetic information, which are very essential for the growth, the development, and the reproduction of organs. RNA, in case it translates the genetic information into proteins by directing the synthesis of polypeptides in the ribosomes. Significance in cellular process, if we say, nucleic acids are fundamental in storage and expression of the genetic information. They guide the synthesis of proteins which perform most of the cell functions. We'll move on to the lipids. Lipids properties, function, and significance. Okay, lipids. Lipids. Did I miss skip the protein? Yeah, protein is there in between. Okay, so proteins are they are the polymers of amino acids which are linked by the peptide bonds. They have 4 levels of structures. The primary structure, that is sequence of amino acids, a secondary structure which is called as alpha helices or beta sheets. Then there is a tertiary 3-dimensional folding structure, and a quaternary, the assembly of multiple polypeptides. The functions of these proteins, the main function including the enzymes, which are nothing but the catalyzing the biochemical reactions, providing the support, mainly the collagen, transporting molecules, mainly the hemoglobin in blood, regulating the processes like hormones like insulin, which is used for the regulation. Significance in cellular processes. Proteins are essential for the virtually every process within the cell, which includes the catalysis, transport, structural support, and communication. So they ensure proper cell functioning and response to the environmental changes. Now let us move on to lipids. Lipids, the properties of lipids, if we say, lipids are hydrophobic molecules composed of mainly carbon and hydrogen. They include triglycerides, which are the fats, include phospholipids, include steroids. Lipids are not polymers but are formed by assembly of similar molecules, not like proteins. Proteins are amino acid polymers. Lipids are not polymers but similar to that, not that much lengthier in size. Functions. Lipids store energy, provide the required insulation and protection, and they form the cell membranes. Triglycerides, they store energy. Phospholipids from the bilayer of cell membranes, and the steroids, which function as hormones. Example, the estrogen and testosterone. Significance in cellular process. Lipids are crucial for the construction of cellular membranes, to protect the cell and control the movement of substances in and out. They also play a role in energy storage and signaling. Now move on to the next topic, that is enzymes. Question is framed. Classify enzymes based on their functions. Provide one example for each class. Elaborate on significance of enzymes in biological reactions. Course outcome 1, level 3, 10 marks. So answer scheme and classification with example carries 5 marks. Elaboration on significance of enzymes carries 5 marks. Enzyme classification. We'll move on to enzyme classification. The first one, that is oxidoreductase. The main function of oxidoreductase is to initiate or to catalyze the oxidation reduction reactions, simultaneous redox reaction we call it as. Okay, so to catalyze the oxidation reduction reactions, oxidoreductase is used as a catalyst, that is as a enzyme. Example, the lactase dehydrogenase. Okay, lactate dehydrogenase, it converts catalyzes the conversion of lactase to pyruvate during the anaerobic the respiration. Second classification, that is transferase based on the function. They transfer the functional groups between molecules. For example, the kinase which transfer phosphate group from ATP, adenosine triphosphate, to proteins, and regulating the cellular process like the signal transduction. Then comes hydrolysis. Hydrolysis, the function is to hydrolyze hydrolysis reactions. Hydrolysis means adding water molecules, breaking down molecules by adding water. Example, lipases, which hydrolyzes triglycerides into fatty acids and glycerol, which is the fat digestion. Okay, then next one, that is lyases. The function of lyases to remove or removal of some groups from the double bonds. Addition or removal of bonds to the double bonds. Unsaturated molecules, double bonded molecules. Okay, so example, pyruvate, covalent bond, single bonds, double bonds, that same organic double bond only mean here. Many organic molecules are having double bonds in their structure, so molecule can be added to that double bond, molecule can be removed from there also. So example, pyruvate decarboxylase, which removes a carbon dioxide molecule from the pyruvate to form acetaldehyde during the alcoholic fermentation. We know aldehyde functional group. Aldehyde is C double bond or single bond H, carbon connected to hydrogen to one side, and carbon containing double bond with oxygen, and again connecting to other carbon containing molecule, that is aldehydes. Okay, so where the double bond involvement is lyases is used. Then comes the isomerases. Isomerases, it catalyzes in rearranging the atoms within a molecule to form the isomers. You know what are isomers? Same copy, similar type of molecules. Okay, so triose phosphate isomerase, it catalyzes the conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate during the glycolysis. Then comes the sixth one, that is ligases. The function of ligases to catalyze the joining of two molecules, usually the ATP, adenosine triphosphate. Example, the DNA ligase, it joins DNA strands together by catalyzing the formation of phosphodigester bonds during the DNA replication and repair. Now comes the elaboration significance of enzymes. Elaboration on significance of enzymes, that is the meaning. Enzymes are very having a vital role in biological reaction due to the many following reasons, like they catalyze. Enzymes accelerate chemical reactions by lowering the activation energy, which is required for the reaction to occur. Okay, if the activation energy is less, then the molecule will react. If it is in higher energy state, activation energy required is high, then that much energy cannot supply, therefore it reduces the activation energy. which allows the biochemical reaction to proceed at the physiological temperatures, making them or enabling the life sustaining process to occur very efficiently. Then specificity, the enzymes, they exhibit a very high specificity for their substrate. The key lock mechanism that we studied, it recognizes the binding and it recognizes and binds to a specific molecule based on their shape as well as the chemical property. So this specificity ensures that enzymes only catalyze only specific reactions and preventing unwanted side reactions and maintaining the required homeostasis in the body. That is the condition required to maintain the physical conditions, temperature or other temperature when compared to the physical environment, the environment, outside environment. Outside environment, if it changes also, the body has to maintain the same condition, that is called homeostasis. Then regulation, enzymes activity can be regulated to control the cellular processes, and they respond to the changing environmental conditions. This regulation mechanism, it includes the allosteric regulations, the covalent modifications, gene expression regulations, which allows the cells to fine tune the enzyme activity as whenever it is needed. Metabolic pathways, uh, metabolic pathways in which the enzyme participate in a series of interconnected biochemical reactions which convert the substrate into a required product. So these pathways, they regulate the process such as the enzyme, the energy production, biosynthesis of macromolecules, degradation of nutrients, which ensures a proper cellular function. Then biological applications widely used in various industries, food production, pharmaceutical, environmental remediation. For example, the use of proteases. It is used in the laundry, that as a detergent. Restriction enzymes in molecular biology is also another example, and the lipases in biodiesel production. These are all some of the very important examples which I can quote. And move on to the next, examine the properties and functions of nucleic acid, uh, emphasizing their role in genetic information storage and transfer. Discuss relevant industry applications, that is post outcome 1, level 3, 10 marks question. 4 plus 4 plus 2 is the mark distribution which I gave. Examination of properties, emphasizing role in genetic information, discussion of industrial applications. So examination of properties and function of nucleic acid, properties of nucleic acid. Nucleic acids are very large, a complex molecule composed of nucleotide monomers, and each that nucleotide, it consists of that DNA, RNA, which I am telling about helical structures. Okay, so the it consists of phosphate group, a sugar which is deoxyribose in DNA, ribose in RNA, and it consists of a nitrogenous base, adenine, thymine, cytosine, guanine, DNA, adenine, uracil, cytosine, guanine in RNA. Nucleic acids, they are having a double stranded helical structure in DNA and a single stranded structure in RNA. The functions of nucleic acid, and we discuss. The main function is to storage of genetic information, the information to be carried to the next generation. DNA, it serves as the primary repository, storage of genetic information in the cells. It carries the instruction which is necessary for the development, the growth and functioning of organisms. Then it is used in a transfer of genetic information. The RNA, which plays a crucial role in transferring the genetic information from DNA to the copying mechanism called as the cellular machinery which is responsible for the protein synthesis. It acts as a messenger RNA, mRNA. It acts as a transfer RNA, tRNA. It also acts as a ribosomal RNA, that is rRNA. Now, emphasizing the role in genetic information storage. DNA stores the genetic information in the form of a linear sequence of nucleotides. The sequence of nucleotides which encodes the instructions from synthesizing the proteins and controlling the cellular process. The genetic information, it is actually passed from parents to the next generation, that offspring, through the DNA replication and inheritance. The genetic information transfer that is during the gene expression. DNA it is transcribed into mRNA, which carries the genetic code to ribosomes. This is when during the information passage, creating the proteins, or during the replication of DNA. DNA strand, double strand, it becomes a one strand, single stranded. A extra copy is formed there, so that it becomes a RNA, single strand becomes an RNA. So DNA, it is transcribed into a messenger RNA. DNA gives a single strand, then we call it as a messenger RNA. So this carries the genetic code to the ribosomes. Ribosome is a machine, a part where the mRNA go and sit, just like the printing ink, or printing whatever called in the printer. Whatever you want to print, that sketch has to be formed. So that DNA forms the mRNA, which is a sketch in the ribosome. Ribosome is the actual printing box, that machine. Okay, there the mRNA sequences into specific amino acid sequences combined there. Ultimately, it forms the proteins. So this process which ensures the accurate transfer of the genetic information from DNA to protein, which is very essential for cellular functioning and other developments. Then industrial applications. So DNA sequencing. Okay, just jumped. I was genetic engineering was missing. So genetic engineering, one of the main industry application. Nucleic acids are used in genetic engineering techniques such as the recombinant DNA technology, which is used to modify organisms for various purposes, which includes the biopharmaceutical production, the agricultural, the environmental remediation, etc. Then comes the DNA sequencing. The industrial application of DNA sequencing, it include medical diagnostics, forensic analysis, biotechnology research. These sequencing technologies have revolutionized the fields such as personalized medicine and the genetic screening. These are the new fields of studies. Next question: analyze the structure of proteins and their diverse functions in living organisms. Provide examples to illustrate the importance of protein in biological processes. CO1, level 3, 10 marks. Structure analysis of protein, 2 marks. Discussion of diverse function, 5 marks. Illustrative examples, 3 marks. Illustrative examples, I am not given the image as such. I am not giving image in this class series. Okay. Now we move on to the answer scheme. Protein structure. Proteins are polymers of amino acids which are linked by the peptide bond. The primary structure that is a very linear sequence of amino acids, linearly arranged amino acids. Secondary structure, that is the folded structure. Okay, the amino acid as it is formed long chain, it will start folding in their own different methods of foldings are there. Folding patterns called as alpha helixes or beta sheets. These are the different folding patterns which we call as the secondary structure of protein. Tertiary structure, that is overall three-dimensional shape of the protein. Three-dimensional structure of protein is tertiary structure. Quaternary structure, arrangement of multiple protein subunits if applicable, that is quaternary structure. Then enzymatic activity. Proteins act as enzymes which catalyzing the biochemical reaction. Example include the digestive enzymes like amylase, metabolic enzymes like ATP synthase. It gives the structural support. Structural support, proteins provide structural support to the cells and tissues. For example, the collagen forms the structural framework of the connecting tissues such as the skin, and the tendons and bones. Then transportation, some proteins they function in transporting molecules across cell membranes throughout the body. For example, in the blood, the hemoglobin is an example of a which transport oxygen in the bloodstream. Then hormonal regulations. Proteins they serve as hormones regulating various physiological processes. Insulin, which is produced in the pancreas, regulates the glucose metabolism and the blood sugar levels. Then immune response, disease response, or the proteins they are very integral to the immune system, acting as antibodies to recognize and neutralize the foreign pathogens or the bacteria, viruses, fungus, etc. They are the pathogens for us, for our body. Okay, so that is, we have to have our own immune system to resist that. So immunoglobulin is an example of such a protein which gives the immune responses. Illustrative examples. Okay, so only example is given, illustration is not there, explanation is there. Digestion, the enzymes like the pepsin, trypsin, okay, these break down food molecules into smaller nutrients for the absorption. Then the muscle contraction, proteins such as the actin and myosin for the elongation and expansion and compression of a muscle contraction, as well as the muscle movements are done with actin and myosin. DNA replication, DNA polymerase is an example of protein enzyme that synthesizes new DNA strands during the DNA replications. That's about the biology for engineers BBOK407 part 2. Thank you. More class videos are coming. Please be tuned in. Please do subscribe, like button, bell button, so that you will get the notification as and when I complete the video class. So once again, thank you very much.